Hello, everyone. My name is Nicole Egan, and today I'm going to be talking about mortality and behavioral effects of both agricultural runoff and imidacloprid and chlorantraniliprole on Daphnia magna. So I'm going to give an overview of what we're going to talk about today. There was two studies that I'm going to talk about. One where we exposed Daphnia magna to field water in the Salinas River watershed and assessed mortality and swimming behavior, as well as had chemical analysis of the field samples. And the second, which we exposed them to just chemicals, which were chlorantraniliprole and imidacloprid, and then assessed their behavioral changes. So for people that don't know, Salinas is in California, and it is a highly productive agricultural region. They make lots of agricultural commodities like strawberries, artichokes, and lettuce. And because of that, there's lots of pesticides being used in this area, lots of different pesticides, and many of them end up in the waterways surrounding them. Because of this, DPR, the Department of Pesticide Regulation in California, has regular monitoring sites, which are marked here in these colored parts here. But we sampled at 3 of these sites, the Salinas River near Davis Road, the Aliso Slough at Hartnell Road here, and the Quail Creek near Highway 101. And when we came back with our field samples, we did dilution series where each, for example, Quail Creek, we came back and each of them had a 100%, meaning that 100% of the treatment was water from Quail Creek. 60%, only 60% of the water was from Quail Creek, and 40% was our control water, 35 and 20% in the same way. And each of these treatments, we had 5 replicates, which each had 10 individuals. So for each treatment, 50 individuals were being exposed to this dilution of the field water. And a good thing to note is that our field samples had both water and some sediment in it. So that way we were able to capture chemicals that preside in the water, as well as chemicals that are going to sorbed to the sediment. And of course we had control groups. So each treatment, Quail Creek, Aliso Slough, and Salinas River, had a control group that we compared it to, which was pH, alkalinity, and hardness tested in order to make sure that they were in the ranges that are preferred by the Daphnia. And for each of these, there was also 5 replicates with 10 individuals. And from there, we did a 96-hour static renewal exposure test with following EPA protocols. All of our organisms were put into temperature and light controlled chambers for 96 hours. We tested mortality every day, and we fed them at the halfway point, and as well as we changed their water at the halfway point as well. And we did all of this to ensure that any sort of endpoints that we were measuring in this study, it was mortality and behavioral changes, were actually due to exposure to the chemicals that were in our samples, nothing else. So no temperature problems. It wasn't that the pH was different. It wasn't that they were having some other sort of stress. It was just chemical stress that was causing these changes. So like I said, we tested mortality at every day, and the most toxic site was Quail Creek. We had 0% survival, even in the 20% dilution. So that means that even though only 20% of the water in this treatment was from that site, and 80% of it was control water, still all of them died. And this was considered to be toxic. Aliso Slough, we also considered it to be toxic because still, even at 20% water from Aliso Slough, 27.5% of them died. So Salinas River, on the other hand, was less toxic. This was to be expected because of the fact that these are more upstream, they're closer to the agricultural region, so more of the chemicals are going directly into these areas, as well as the fact that there's less water here, so they're going to be at a higher concentration. Whereas the Salinas River, lots of things end up flowing into the Salinas River. There's more water, so it's going to be more diluted. There's going to be a lower concentration, and so it makes sense that this was non-toxic. In addition, our control group, we had 100% survival, so no one died, which was a good thing. So now moving on to swimming behavior, this graph can be a little bit confusing to look at, so let me just walk you through it real quick. So we tested total distance moved of our organisms, and we tested it over 30 minutes. And the box here represents a 10-minute period of dark. So we tested how they were swimming in the dark. We turned on the light, saw how they responded to the light, which is an important thing to look at here, is when the light turns on, how do they respond? Because this is showing how do they respond to changes in their environment, and how does that differ after being exposed to a chemical. And then here we had the latter 20 minutes in the light. So you can see here that the gray is the Salinas River, and this had the most divergent swimming pattern. After the light turned on, it was hyperactive, meaning that it was swimming much more than the control group was in comparison to the Elysial Slough and both the control group, actually. And in addition, its response to the light stimulus was much less. So the light turned on, and it didn't have a drop in swimming, the amount of swimming that it was doing in comparison to the control group here. Here we saw, this is the 20% dilutions. Oh, something that I forgot to mention, sorry. Both of these, Quail Creek is not included because, as I said, there was 100% mortality, so we had no organisms to do swimming behavior tests on. It's the same here as well. But both the Elysial Slough here and the Salinas River were divergent from our control. So in the dark, the Elysial Slough was hypoactive, meaning that it was swimming less than the control group, both in the dark and the light, whereas the Salinas River, the same as before, was hyperactive once the light turned on. And both of these here had a lessened response to a light stimulus in comparison to our control group. So something that you might be thinking, they were exposed to this field water, but what was actually in the field water, what could be doing this, that's making these changes in their swimming behavior? So Department of Pesticide Regulation helped us in this study and did the analytical chemistry. And as I mentioned before, the Salinas River had the lowest amount of chemicals. These NDs means no detections, so it was at a level that was below detectable for the machines that they were using. And this chart here is a very, it's only a small subsample of the amount of chemicals that were found in the sites that we looked at. I want to stress this. This is not all the chemicals that were found. This is just to illustrate that there is a large range of chemicals that are being found in these samples. And you may be wondering why these two are highlighted. That's because chlorantraniliprole and imidacloprid are chemicals of interest to the DPR, and they were found across all three of our sites. So from this data, we decided that we wanted to do a follow-up study where we looked at the behavioral changes in Daphnia magna after being exposed to chlorantraniliprole and imidacloprid by themselves, not with all these other chemicals included. It's important to note that the Quail Creek had the highest mortality. This did not have the highest levels of imidacloprid and chlorantraniliprole, but mortality isn't really what we're looking at here. Like, I know that might seem a little weird. You're like, why would you look at these? These didn't even have the highest concentrations in Quail Creek. But we're trying to look at how these affect behavior. So we're trying to look at a sublethal effect, meaning an effect that happens at concentrations that are below lethal. So we did the second study where we exposed the Daphnia to environmentally relevant levels of IMI and CHL, IMI being imidacloprid and CHL being chlorantraniliprole. And we did this based on the field detection ranges, EC and LC50 values of both of these chemicals, and previous binary and single chemical exposures that was done in our lab. And we came up with 1 microgram per liter for both the chemicals of the low exposure and 5 micrograms per liter for the high. So we followed a very similar method as we did for the previous exposure. There was no dilution series. We just had the low and the high, but the 96-hour exposure was pretty much the same and followed EPA protocols, and we had the same control groups as before as well. And this is the data that we had for the behavior changes for Daphnia magna exposed to chlorantraniliprole after 96 hours. What I really want to point out here is the same as before. The first 10 minutes is in the dark. the last 20 minutes is in the light, and this line here shows where the light is turning on. And you can see that the blue control group has a very large dip in movement. So the light turns on and they do not move nearly as much as they were before. But the organisms that were exposed to CHL had a lesser response here, as well as a sort of delayed response. They did go down, but it didn't go down very much until around here, whereas it really went down here for the control group. So here we are seeing a lessened and delayed response to light stimulus in Daphnia magna that were exposed to both low and high levels of chlorantraniliprole. Imidacloprid, on the other hand, in the chlorantraniliprole, you can see that the amount of movement totally wasn't that different. But here, the control group is moving much more than those that were exposed to the imidacloprid. The imidacloprid treatments were hypoactive in comparison. They moved much less, both the low and the high concentration. And when you're looking at the response to light, there is a delayed response here as well. So at first, both in the high and the low, there is actually an increase. There is an increase in movement and then a decrease. So they first increase, then decrease, which definitely is not seen when there is the light in the controls. So this is something that is important for how this exposure and change to light stimulus is important when looking at how this is going to change them in the wild and the environment. And I'll get into that in a minute. So what are the important takeaways from this? Why is it important that we did this? And what's the reason that you spent time and money on it? The first thing to take away is that these surface waters can have large amounts of chemicals in them. This is mostly going back to that analytical chemistry portion that I was talking about. There can be chemicals from wastewater effluent, agricultural runoff, which is what I've been talking about mostly, industrial waste, like companies dumping, residential runoff, and more things. So there's lots of different things coming from lots of different places that are in this water. And animals live there. So it's important for us to think about how these different chemicals can interact with each other and how many chemicals are really in the water that they're living in. Now, this is really important because these chemicals can cause physiological effects via chemical stress. So when these chemicals are in the water, they cause chemical stress, and these lead to physiological changes that decrease the biological fitness of an organism. This then leads to a change in behavior, less swimming or more swimming, and also really important is that change in how they react to changes in their environment. So like that light stimulus, that can affect things like how they find mates, how easily they can get away from predators, how they can find food, and those behavior changes could have population level effects where they can decrease the population size because they're having less children because they're being less successful in the environment that they're living in. There's an environmental mismatch, right? They are not properly equipped to live where they are living. And this can lead to ecosystem level effects because, as most people know, the effect of one population can have effects on other populations. And these ecosystem level effects are what us humans really count on and what we really care about, because that can have monetary effects. It can hurt things that we like to hang out in, like the sloughs and things that we think are so beautiful. We want to be able to keep that biodiversity. So these behavior changes are really important and sensitive endpoints to be looking at in order to try and predict where those ecosystem level effects are, even if the levels of chemical are lower than what would immediately kill an animal. So I want to thank everyone for listening to my talk, but I especially want to thank my PI, Richard Conan from the Conan Lab, Dr. Shin from the California DPR, the Department of Anatomy, Physiology, and Cell Bio at UC Davis Vet Med, and special thanks to Sarah Simpson for everything she did. She really helped me with all this learning, all this stuff, with letting me help with her projects, and even letting me do my own projects. So I want to thank everyone, and I hope everyone's safe, and you have a good day. Thank you.